Hello dear students, welcome to a new session of Bioprime Points. In today's video, we are going to discuss a chapter called Applications in Biotechnology. First prime point is the applications of biotechnology in the field of agriculture. So in agriculture, there can be three different types of patterns of agriculture. One is called agrochemical based agriculture, then organic agriculture, the third one, genetically engineered crop-based agriculture. Three mechanisms: agrochemical, then organic, then genetically engineered crop. These are the three methods. The Green Revolution succeeded in increasing the yield of crops mainly due to use of improved varieties of crops and use of agrochemicals, that means fertilizers and pesticides. The use of genetically modified plants has been useful in the following ways. Genetic modification has made the crop tolerant to abiotic stresses like cold, heat, drought, salinity, etc. Then it has reduced the dependence of crops on chemical pesticide as they are made pest resistant. And due to increased efficiency of mineral usage by plants, early exhaustion of fertility of the soil is prevented. The fourth one, the post-harvest losses are much reduced. So these are the significances of using genetically modified plant-based agriculture. Okay. Then, applications of biotechnology in the field of medicine. The recombinant DNA technology has been used in the production of safe and effective therapeutic drugs. The recombinant therapeutics do not induce unwanted immunological responses that are commonly observed with similar products isolated from non-human sources. Okay, so these are the applications in the field of agriculture and in the field of medicine. A very important application in the field of agriculture is the production of Bt cotton. Bt cotton means a cotton plant having genes taken from a bacterium called Bacillus thuringiensis. The soil bacterium Bacillus thuringiensis produces crystal proteins called cryproteins that are toxic to larvae of insects like tobacco budworms, armyworms, beetles, and mosquitoes. But the cryproteins exist as inactive protoxins and get converted into active toxin when ingested by the insect, as the alkaline pH of the gut solubilizes the crystals. The activated toxin binds to the surface of the epithelial cells of the midgut and create pores. This causes swelling and lysis of the cells, leading to the death of the insect. The genes, that mean the cry genes, encoding this protein are isolated from the bacterium and incorporated into several crop plants like cotton, tomato, corn, rice, soybean, etc. Okay, but since the toxin is existing inside the body of these plants as an inactive form, there is no toxic effect to the plants. But when this protoxin get activated inside the insect pest due to the alkaline pH of the gut, then only the toxicity will be expressed. For example, cotton bollworms that can be prevented by the cry genes, cry1AC and cry2AB. Corn borer can be prevented by using cry1AB. Colorado potato beetle can be prevented by using cry3AB. And corn rootworm can be prevented by using cry3BB. Okay. Another aspect of biotechnology in the agriculture field is a phenomenon called RNA interference or mRNA silencing. A nematode called Meloidigain incognitia, it's a round worm that infects tobacco plants and reduces their yield. The specific genes in the form of cDNA means copy DNA, from the parasite are introduced into the plant using agrobacterium as a vector. The genes are introduced in such a way that both sense strand and anti-sense strand are produced. Sense strand means the strand that is capable of producing the RNA, and anti-sense means that is not producing the coding strand. So these are complementary to each other. Since these two RNA are complementary, they form a double-stranded RNA, that is dsRNA. This neutralizes specific RNA of the nematode by a process called RNA interference. In the central dogma, we studied that DNA is DS, double-stranded. It produces single-stranded RNA, and only from this single-stranded RNA, the protein can be produced by a process called translation. But when the RNA itself is made in a double-stranded form, the translation is not possible, isn't it? That means the functioning of the RNA is silenced. So that is why it is called mRNA silencing. The parasite cannot live in the transgenic host, and thus the transgenic plant get protected. Okay, this process is called RNA interference or mRNA silencing. Then another important biotechnological application in the field of medicine is the production of genetically engineered insulin. The human insulin consists of two short polypeptide chains, chain A and chain B, which are connected by disulfide bridges. That is an active form of insulin. But insulin is secreted as a prohormone, which has to be processed before it becomes functional hormone. At the time of secretion, insulin consists of three chains, chain A, B and C. That is a prohormone, inactive hormone. The prohormone contains another polypeptide called C peptide, which is removed during maturation. So in our body, ABC condition is produced, and during maturation C will be removed and the AB form the functional insulin. Okay. So in 1983, Eli Lilly, an American company, prepared the two DNA sequences, that means chain A and chain B separately, and they integrated together forming functional insulin. In the two DNA sequences coding for chain A and B of the human insulin, and introduced them into plasmids of E. coli to produce insulin directly. The two chains produced were extracted and combined by creating disulfide bridges. So this is how a functional insulin can be created, and that insulin is called genetically engineered insulin. Okay. Another prime point is called gene therapy. Genes are inserted into cells of an individual to correct certain hereditary diseases, genetic diseases. Viruses which attack the host and introduce the genetic material are used as the vector. The first clinical gene therapy was given in the year 1900 to a 4-year-old girl with the adenosine deaminase deficiency. It is also called ADA deficiency. That can be cured by bone marrow transplantation in some children, but it is not completely curative. For gene therapy, lymphocytes were grown in a culture and functional ADA-cDNA, which is a copy DNA, is introduced into the lymphocytes. If a functional gene is introduced into the bone marrow cells at an early embryonic stage, it would be a permanent cure. That means a genetic disorder can also be treated to some extent by using biotechnological application. One such example is called ADA deficiency. Then another one is called molecular diagnosis. That is also an application of biotechnology in the field of medicine. The recombinant DNA molecules and techniques like PCR are used for early diagnosis of disorders. The cloned genes, when expressed to produce recombinant protein, help in developing sensitive diagnostic techniques like ELISA. ELISA means enzyme-linked immunosorbent assay. The cloned genes are also used as probes to detect the presence of a complementary DNA strand. The presence of a normal or mutant gene can be detected in such method. Then PCR, that is the polymerase chain reaction that we already studied, is used to detect the HIV and to detect mutations in genes. That means the early detection of a disease can be diagnosed by using techniques of biotechnology. Okay. Another prime point is the transgenic animals. Transgenic animals are those that have had their DNA manipulated to possess or express a foreign gene. The gene that's transferred into an organism is called transgene, and the process is called transgenesis. An animal that's created is called transgenic animal. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions and the body and its development. Transgenic animals are designed to increase our understanding of how genes contribute to the development of diseases, and they are made to serve as model for various human diseases. The transgenic animals that produce useful biological compounds can be created by introducing a portion of DNA that code for that product from other organisms. Example, alpha-1 antitrypsin is used to treat a disease called emphysema. It's a smoker's disease. The first transgenic cow, Rosie, produced a human protein-enriched milk. It also contained a human alpha-lactalbumin, a more nutritionally balanced product for human babies. Then transgenic mice are being developed for use in testing safety of vaccine. Example, polio vaccine. Transgenic animals with more sensitivity to toxic substances are being developed to test toxicity of drugs. So all these are the applications of biotechnology by using transgenic animals. Okay. The next point, even though biotechnology has so many beneficial applications, some ethical issues are also there. What are the ethical issues? The genetic modification of organisms can have unpredictable or undesirable effects when such organisms are introduced into an ecosystem. The modification and use of such organisms for public services has also resulted in problems with the granting of patents. Hence, Indian government has set up an organization which is authorized to make decisions regarding the validity of the genetic modifications and the safety of introducing genetically modified organisms for public services. That organization is called GEAC, Genetic Engineering Approval Committee. Then another important point also is there, that is called biopiracy. What is biopiracy? The usage of bioresources and traditional knowledge of other countries without proper authorization or compensation is called biopiracy. Example, basmati rice grown in India is distinct for its unique flavor and aroma. But an American company got patent rights on basmati through US Patent and Trademark Office. The new variety of basmati has been developed by this company by crossing an Indian variety with a semi-dwarf variety. Okay, so that process is called biopiracy. These are the negative aspects of biotechnological developments. Some ethical issues are there, then piratic activities are also there. Okay, so these are the prime points of the second chapter, applications of biotechnology. Thank you.